Tutorial: Pooling of human stool samples. In this tutorial we will demonstrate how to pool human stool samples for rapid assessment of both intensity of soil-transmitted helminth infections as well as drug efficacy. 1. Equipment. When you want to pool stool samples, the following equipment is necessary: 10 plastic beakers of at least 60 milliliters, a scale, a permanent marker and a stirring device such as a tongue depressor. 2. Pooling procedure. General format. The pooling procedure we will present is developed to obtain different pool sizes with a minimal quantity of stool per individual. An example will clarify the procedure. Assume that we have 60 individual stool samples, here indicated in yellow, and these are organized in 6 rows of 10 individual stool samples. Subsequently, we will transfer a fixed quantity of stool from each of the 10 individual samples of one row into a new beaker, which then results in 6 pools of 10 individual stool samples, here indicated in green. Afterwards, we can use these pools of 10 to make larger pools. To do this, we will transfer a fixed quantity of each pair of pools of 10 into a new beaker. In that way, we make 3 pools of 20 individual samples, here indicated in red. Analogously, we will transfer a fixed quantity of the pools of 20 into a new container to obtain 1 pool, which then represents 60 individual samples, here indicated in blue. We will now show the procedure more in detail for pools of 10, 20 and 60 individual stool samples separately. Pools of 10 individual stool samples. Organize 60 individual stool samples into 6 rows of 10 stool samples. This can be done either randomly or by stratifying the stool samples in rows. For example, all stool samples provided by subjects within the same age range are organized in one row. In this example, the samples have been organized randomly. Label a plastic beaker and put it on the scale. Press the tare key and transfer exactly 1 gram of each stool sample of one row to the rim of this pre-labeled beaker. To avoid pooling the same sample twice, put the stool sample aside once it has been added to the pool. Repeat the procedure for the 9 remaining stool samples. At the end of this step, the balance should display a total mass of 10 grams and 10 samples should be placed aside. Subsequently, homogenize the stool samples thoroughly with a stirring device. It's not always clear when to decide a sample is entirely homogenized. However, stool samples can vary considerably in color, and hence, as a rule of thumb, it can be stated that a pool is thoroughly homogenized when it is equally colored. Next, repeat the preceding procedures for the 5 remaining rows to obtain 6 pools of 10 individual samples. Finally, each of these pools can now be processed with a diagnostic method. Pools of 20 individual stool samples. Organize 6 pools of 10 individual samples into pairs. Label a new plastic container for each pair of pools of 10. Press the tare key and transfer 5 grams of each of the 2 pools to the rim of this pre-labeled beaker. Thoroughly homogenize the stool samples with a stirring device. Repeat the preceding procedure for the 2 remaining pairs of pools of 10 to obtain 3 pools of 20 individual samples. Each of these pools can now be processed with a diagnostic method. Pools of 60 individual stool samples. Label a new plastic container and place on the scale. Press the Tare key and transfer 3.3 grams of each of the 3 pools of 10 to the rim of this pre-labeled beaker. Thoroughly homogenize the stool samples with a stirring device. The pool can now be processed with the diagnostic method. 3. Acknowledgements. The raw material for this tutorial was collected during a field study, financially supported by the Institutional University Cooperation Project of the Flemish Inter-University Council. The Research Foundation Flanders supported the development of the tutorial through a postdoctoral mandate grant.